Hi everyone. It's a big responsibility to be opening this session. So I will try to do my best to give you an overview of our activities here at the Swiss Bee Research Center. So we are located in Bern, Switzerland. And as there is a rising interest in Switzerland as well in the field of conserving the native honeybee, there have been a lot of activities at the Swiss Bee Research Center in this direction in the last years. So we have been hosting a PhD thesis. Gabriel Zahn has been here. Melanie Pacheco, many of you may know her, has also been studying here. And so we are still having projects in this area. And today I'm going to present an overview of what I've done myself in this year, and also my colleague, Dr. Annick Gmel, also from the team of Markus Neuditschko. She has also been a part of this project. So I'm going to summarize everything to give you an overview of the situation here. So first of all, for those who are not familiar with beekeeping or local subspecies in Switzerland, so the dark honeybee Apis mellifera originally is a native subspecies of Apis mellifera in the region north of the Alps, so which encompasses the biggest part of the Swiss territory. South of the Alps, it's very likely that we had Apis mellifera ligustica, but some previous work by Melanie Parejo was not able to find really pure Italian honeybees in this region anymore. So the purple region was Ticino, which is south of the Alps, close to northern Italy, but where apparently this subspecies is not present anymore at a pure stage. But even north of the Alps, the situation of the dark honeybee is not so secure, because starting in the 19th century and accelerating in the second half of the 20th century, there have been many imports of Apis mellifera carnica, and later also by GWAS strains, which have been selected in other European countries. And because there have been more and more of these imports, this situation led to uncontrolled matings with admixture and probably also a loss of local adaptation. So the situation has been recognized by the beekeepers, who try to organize themselves to save the dark honeybee. So the association mellifera.ch had a leading role in this regard. And I'm sure you will hear more about this topic with the presentation of Matthieu Guichard in a few weeks. So this is the current challenge for Apis mellifera in Switzerland. And the current situation, it's a bit complicated. Currently, there are two areas in Switzerland which are recognized by law as conservation area. So this is the case for the canton of Glarus, where there is the oldest legal protection of the dark honeybee. It's only allowed to rear Apis mellifera. There is another smaller area which is called Melchtal, a bit closer to the west of Switzerland, but there it's much smaller. There have been also two attempts to create new conservation areas in the eastern part of Switzerland, as well as in the Bernese Oberland. But as Matthieu Guichard will tell you, it has been very complicated, and currently the projects were not able to succeed because of opposition of single beekeepers. So this is for the conservation part, which means areas where Apis mellifera can still survive just by natural matings. Of course, there is still a proportion of hybridization, but beekeepers try to take care of their dark honeybees. And in parallel to this project, there is also a selection program who aims to provide dark honeybees also to beekeepers located out of the conservation areas. So currently, the selection program takes advantage of 5 mating stations and provides queens for testing networks. So they're evaluating colonies each year in order to select the best colonies for drone production at these mating stations. For those who are not familiar with Swiss beekeeping, in Switzerland, we don't have islands like in other regions. So to produce queens, the best way to have big quantities of queens produced is to work with these mountain mating stations, like in Austria, for instance. So they're always located in insulated valleys where drone-producing colonies are put, and beekeepers go there with their queens to have them mated. So the idea is that the reliability of the mating stations are dependent on the isolation of the valleys. So here in this example, the valley is quite remote. So we can suppose that a very small proportion of foreign drones are able to go in. And the most common situation is that in every mating station, there is one paternal origin, which means that all colonies providing the drones are all led by sister queens. There have also been attempts in the last years to work more on pool mating stations, which means the drone producing colonies are not necessarily unrelated, but can also belong to diverse lineages, all showing very good aptitudes for beekeeping. But in the case of the data, which I will present later, we had a situation where all the queens leading the drone-producing colonies in the mating stations were sisters. So this is what I represented on the map. So for each mating station, we have one paternal origin, which here I numbered from P1 to P5. So currently in Switzerland, you may have understood the current challenges. So we have challenges as well for the conservation part, but also for the selection part. Starting with the selection part, current challenge is to identify traits and also genes associated with better colony survival, for instance, when the colonies are facing our destructor. But in order to promote Apis mellifera also to beekeepers, the honeybees produced by selection also need to have a positive ability to be used by beekeepers, which means not to be too aggressive, not to swarm too often, and also to provide a honey crop for beekeepers. In parallel, it's interesting for both the selection program and the conservation program to monitor genetic diversity, to monitor inbreeding, and to monitor admixture, in order to evaluate the sustainability of the population and to what extent the population is threatened either by incoming of unwanted genes or just by a lack of genetic diversity in the population. So to try to give an overview of the current situation, we have performed 2 studies, but both are based on whole genome analysis, and they can provide useful tools for population management of the dark honeybee in Switzerland. So in order to identify the best traits and the best associated genes favoring colony survival, we have performed genome-wide association studies, which are called GWAS. And also, in order to monitor genetic diversity, inbreeding, and so on, we performed a diversity study. So I have been part of one of these studies, and the other one has been performed by Annick Kmel. So we have been working for both studies with pools of workers as samples. Why did we do that? If we look at a honeybee colony, a colony is composed in summer of about 20,000 workers, which are involved of most of the tasks of the colony, which are brood care, food supplies, homeostasis, and so on. We have one queen which lays eggs and produces pheromone to have the colony as one functional unit. And we have about 1,000 drones which mate to the queen and transfer the genes to the colonies in the neighborhood. So queen and workers are both diploid, and both of them highly contribute to the phenotype of the colony for most of the traits useful for selection. So just sampling drones is very cheap. You can just have to collect the drones in the field. It's very fast. You don't kill any sexuals, even if there have been a lot of improvement in sequencing also of material from the queen without killing the queen. There have been work in Germany on this topic. It's not very easy for beekeepers to take this material. And by taking also a pool of workers, its advantage is also to represent the genetic diversity within the colony. So if genes are only present in one fraction of the workers, for instance, because they've been mated to a very specific pathway line, we hope that we can also have this in our genomic data. So for both analyses, which I'm going to present to you, we have been working mainly on Apis mellifera mellifera, but as an outgroup, we also decided to include some Apis mellifera carnica. So we have been mainly working with Swiss honeybees. So from both the selection program in Switzerland, but also from one conservation area, the canton of Paris. In frame of a collaboration with our French colleagues from the Beestrong consortium, we have been able to include also some samples from the French conservation area from Savoie. There have already been some exchanges in the last years, also in the frame of the thesis of Melanie Parejo. So it's Savoie is a region of France which is quite close to Switzerland, so it's interesting to compare the situation in both areas and to see if results obtained in Switzerland can also be generalized to other regions. And for Carnica, we took some samples from diverse breeding lines from Switzerland, but also from other countries to be used as an outgroup. So from each colony, we went to the field to take pools of 500 workers per colony. So this number has been chosen by our colleagues in Toulouse in the frame of our Beestrong consortium to represent the diversity within the colony, also to limit the influence of single workers, which could be a bit bigger, a bit smaller on the final result. So the pool of workers was kept frozen after the field and taken back to the laboratory. All the bees were mixed in a kind of honeybee soup, and this pool was sequenced. So at the end, we had one pool for each colony present here in our analysis. So for the next steps of the method, these steps are perhaps interesting for people involved with genetics. You don't need to remember precisely everything which we did. I just give this as an information. So we extracted the DNA. We sequenced the pool for each colony. So this has been done at the INRA at Toulouse with our French partners on the platforms. We aligned the sequences on reference genome AML HAV 3.1. So this is not an Apis mellifera mellifera genome. We have one Apis mellifera mellifera assembly which is currently prepared, but it has not been annotated so far. So we decided to work with this other genome, which has much more annotation information. So based on our pool, we were able to have sequencing depth data, also allelic frequency from the pool, which means if we had 2/3 of A at a certain locus and 1/3 of T at a certain locus, we were able to quantify the quantity of each allele in our colony. So this was done for 7 million SNPs, which had been predefined by our French partners based on previous analysis to cover the whole genome of Apis mellifera. Later, we, based on these 7 million SNPs, which were defined in France, based on that data, we did a quality filtering to remove all loci which had no diversity or multiple alternative alleles, which had too high or too low sequencing depths, and so on. So this was more quality filtering to refine the quantity of data really useful for our study. And we ended up with about 1 million SNPs, which were used for this analysis. We used dosage conversion to convert our allelic frequency data to genotypes. We used diverse options to do this, and you can refer to our publication to have more detail to this point. We had a first look at the population structure. This was done to remove outliers for the analysis, so which could be, for instance, dark honeybee colonies, which in fact were highly hybridized. As genomic results are only valid within one defined populations, it's not easy to have them generalized. So we first tried really to focus on a population which was homogeneous. We performed the genome-wide association study itself. So we associated the genome with the phenotype measured in the field for several traits. So we had population size, honey, pollen stores, gentleness, calmness, recapping, Varroa infestations, and also hygienic behavior towards killed brood. We mapped the genes on the reference genome. and I'm going to present the results. So first, the population structure. So just based on this dosage data, we were able to discriminate the different colonies from our sample quite easily. So just based on the two first principal components of our principal component analysis, we were able to distinguish all the Apis mellifera melliferas, so Swiss selected population, conservation area from Switzerland, conservation area from France, from Vulcanica colonies, which have a gray dots here. But we also identified 5 colonies which were supposed to be Apis mellifera mellifera, which clustered quite tightly with Vulcanica colonies. And these colonies, which are probably highly mixed individuals, were removed from the following GWAS analysis. So, in fact, the most interesting result for GWAS analysis, which we had, was for recapping of mite-infested brood. Recapping is a trait which is currently gaining interest in the field of analyzing traits enabling resistance against Varroa destructor. So honeybees performing recapping open brood cells and can later recap it. But it's supposed that just having this opening and reclosing can disturb mites present in the cells in the reproductive cycle. So it's a trait which is gaining interest because it doesn't imply sacrifice of the honeybee brood. So it may be less costly to the colony than, for instance, a VSH, which is based on the complete removal of infested brood cell. So we had this trait in our data set, and it was a trait which gave the most interesting results. So I'm going to detail this result now. So this is a plot from the GWAS, genome-wide association study. So on the x-axis, you have all the chromosomes, and each single dot is one SNP which we analyzed. So we had 1.4 million SNPs at the end, so we have 1.4 million little dots. And on the y-axis, you have a probability that this dot has a significant impact on the phenotype of the colony. And in red, this is the Bonferroni corrected significance threshold for these traits. So we had 2 SNPs which were significantly associated with the phenotype. So the best associated SNP was located on chromosome 5. This is a small black dot. You may have difficulties to see it, but it's this one. When we look at this SNP, it is a SNP where you have either A or T in your populations. And when you have a percentage of the A allele which is above 50%, so you have 50 to 100% of the A allele in your pools, you have a higher probability to have more cells which are infested, which have been recapped by the honeybees. And the opposite, if you have a lower percentage of the A allele, you have also a lower frequency of recapping of mite-infested cells. And this is significant. What is also interesting is that the percentage of A allele is highly variable between the subgroups we included to our study. So the highest frequency was, in fact, found in the Vulcanica groups. We had a lower proportion of the A allele in the mellifera from Switzerland, and the lowest was in the conservation area. But this could be also interesting because this SNP could also help to distinguish the different subpopulations from each other. Going forward to the analysis, the best associated SNP on chromosome 5 was not located precisely in a gene, but it was very close to 2 genes, which are really interesting. The first one is a gene which is known to be associated with the nervous system of the honeybee. It has been associated with grooming, as well in Apis mellifera as in Apis cerana, the Asian honeybee. One other gene is involved with signaling pathways of the honeybee, which are also signal transduction in the nervous system of the honeybee. So it looks like there have been several genes related to the nervous system and also some other resistance traits in this area. So it's possible that we have here a gene region associated with social immunity, which could be grooming, which could also be recapping. The second best associated SNP, which I've not mentioned before, but which was located on chromosome 4, also had a significant effect on the phenotype. And it was located within one gene, which is named this gamma. And this gamma is also quite interesting because it's involved in neuronal development, and it has been shown to be differently expressed in VSH adult bees compared to control bees. And it's also regulated by mite infestation in brood. So there are quite interesting clues that here there is also something going on with regards to resistance and to recapping. Of course, our dataset is quite small. We have about 250 colonies as a total. This is very small. The results should be verified based on bigger datasets and also in the frame of overpopulations. We did the same analysis for all the traits which I've listed before. We found also QTL associated with gentleness and calmness. For this, we have already published something in Animal Genetics this year. So you can have a look to this publication as well. Sadly, we didn't find any other QTL identified for Varroa destructor-related resistance traits, for instance, mite infestation levels or hygienic behavior towards the brood. We know that some other data from other populations show some QTL for these traits, but we didn't find any, either because our data set was too small or because the considered population has some very specific trait determinism. So for the recapping story, we have a publication which has recently been accepted to Animal Genetics, and we hope it will be published within the next weeks. So let's move on to a diversity study. So the methods are mainly the same as presented before. So we went through the genomic analysis, sequencing, alignment, creative filtering, and so on. The next step are a bit different. So we had for this study to imputate genotypes missing after conversion, because otherwise it would remove a lot of useful information. You would have to exclude some colonies which would have a bad sequencing depth, for instance. So we had to use this method. We calculated admixture levels. We calculated runs of homozygosity as well. I'm going to detail this last point. It is a new method currently used in other species. So runs of homozygosity are quite interesting for genomic studies. We identify regions where you have no variation. For instance, here you have a sequence where you have absolutely no variation. And what is interesting is that this runs of homozygosity, also named rho, can be used to calculate inbreeding coefficients of individuals. So for this, you calculate the frequency of the runs of homozygosity by dividing the total length of these runs of homozygosity by the autosomal length of your individuals. And this gives you this frequency, which is the inbreeding coefficient of an individual. So what we wanted to do is to calculate also this runs of homozygosity for honeybees based on this pool data, which has not been done before. So here are some results. First, the population structure, now based on the whole data set with this method. So it differs from what I have presented the PCA just before, because the PCA was just the two first components. Here, with this method, it allows really to combine every genomic data to calculate genetic distance between individuals. And here on this figure, the thicker the line is, the closer related are the individuals. And the size of the dot, each dot represents one colony, gives also the frequency of runs of homozygosity, which means the inbreeding level of the colony. What is interesting is, as in the PCA, we are able to quite easily discriminate the different subspecies, which are Carnica here at the top, and Apis mellifera here at the bottom, as well as some admixed individuals in the middle. And also interesting is that within the mellifera colonies, we're able to discriminate the conservation area here in dark brown. But also at the mating station level, we're able to discriminate the paternal origins used for the matings. So here you can see that it clusters quite nicely. For instance, here the the light brown colonies all belong, the queens of these colonies have all been mated at the same mating station. And in between, here in light green, we have, I don't see my mouse anymore. Here we have also colonies which are highly admixed and which mainly belong to the French conservation area. So this method seems to be interesting to discriminate subspecies and even within the subspecies level, different mellifera origins. Let's move on to the admixture plot. So this plot, the aim is here to show how related the different colonies are to each other, if there are some origins which are more stable, or are they all a bit overlapping and also admixing with Arcanica colonies. So we used the lowest cross-validation error, which was here obtained with 5 clusters here on the k = 5 line. And what you see is that we have some groups, which are here grouped by a paternal origin on the mating station, where we have a lot of variation within the colonies, but we also have one group here, which corresponds to the Swiss conservation area, where all the colonies are very closely related, very homogeneous. What you see is when we calculate these different clusters, we first clearly distinguish the melliferas from the Carnicas, which happens here with 2 clusters, k = 2 on the first line. We have all the mellifera here quite clearly, and then all the Carnicas. And when we increase the number of clusters until this 5, we can also visualize the structure within the different subpopulations. So we have a homogeneity of colonies, which is very variable among subgroups. What is also interesting is to compare the admixture results, which I've just presented, with the FRO, the inbreeding results, because there is a quite nice correspondence between these 2 types of data. In this tab, you have again the 4 different groups. So the selected mellifera from Switzerland, the conserved mellifera from Switzerland, the conserved mellifera from France, and the Carnica from the various origins. Here we have our number of colonies and also the proportion of the number of colonies which have an admixture which is above 5%. So colonies which can be considered as really admixed. What is interesting is that we have quite a lot of colonies in proportion which are highly admixed in the French conservation area, 13 out of 21. And these are also the colonies which show the lowest runs of homozygosity, which means the lowest inbreeding coefficients. The Swiss conservation area here has no colonies above 5% of admixture, which seems to indicate that the colonies are still quite pure Apis mellifera mellifera. And also, interestingly, we have quite higher runs of homozygosity, which means some higher levels of inbreeding. In the selection program, we're a bit in between, with colonies with a low run of homozygosity level, but also some which are a bit higher. What was a bit surprising is that in the Carnica colonies, which we took from different origins, we had in general also a quite high level of inbreeding, which was really higher than the mellifera colonies. And for now, we don't have any explanation why this is happening. So pool sequence data can be useful to really identify fine-scale structure of honeybee populations, to calculate runs of homozygosity and to deduce inbreeding levels from this. But this is only one type of genomic study. It could be compared with other approaches to see if it can be validated, as calculating runs of homozygosity in honeybees is really a new method. We have to see if it makes sense in this regard, and to compare with other types of approaches. So inbreeding depends on groups. It seems to be very high in Carnica, even with different origins, and the lowest is obtained for the French conservation area. So the interesting thing is that correspondence between inbreeding and admixture results seems to indicate that lower inbreeding is due to higher admixture. This can be a mean to control the degree of foreign drones entering mating areas. If you have more foreign drones, this may increase your admixture and in the same way decrease the inbreeding level of your population. So it may be a means to verify the reliability of the mating stations. So this monitoring should be pursued in Apis mellifera. So if inbreeding increases too highly, it can be the sign of an approaching bottleneck, which could be a threat for the genetic diversity of the honeybee population. If the inbreeding decreases in the next years, it could be a sign that the mating stations are not secure enough, that you have foreign drones entering the mating area. And this could also be a signal to be included in by the beekeepers to perhaps have better protected mating stations, or also to favor political decisions to protect really the mating stations. In the Bernese Oberland here in Switzerland, there are no laws to protect the mating stations. So one mating station, mellifera.ch, is also benefiting from this to really protect the mating zone. If we have a problem with this admixture in the future, implementation of instrumental insemination could improve to ascertain the mating purity. This is also an approach which is followed by the beekeeping organization, mellifera.ch. And obviously, the same approach can be useful to monitor other honeybee populations. So Apis mellifera, but also some native subspecies in other regions. And it could be also interesting to compare between different mellifera origins, for instance, to the UK or in Norway or so on, to see if we obtain also the same type of results. So on this diversity study, which has been mainly prepared by Annick Gmel, we have also a publication in preprint. So general conclusion now. So whole genome sequences provide some important information to evaluate the current stage of conserved or selected honeybee populations, with example here of Apis mellifera. Also, the marker-assisted selection part, it should be applied to the honeybees to have selection precisely for some traits, for instance, resistance against Varroa destructor. And launching these selection programs based on the markers identified here could help to first check the results and later see if we can increase resistance or at least expression of the traits in the honeybee populations. So yeah, this should be continued in the next years. So whole genome sequences should be evaluated for genomic selection, but this is also a political decision. Are the beekeepers ready to have a colony genotyped, to have selection for the whole genome, not only for some markers, but also for the whole genome data? And there will be also some challenges with creating data banks to have all this genome data present and available for selection. As previously mentioned, also the conservation could take advantage of genome studies and admixture estimations to monitor colonies present in the conservation areas, to remove colonies which would be admixed, but also to see if there is a need to have exchange with other conservation areas in order to keep a sufficient genetic diversity, for instance. Yeah, but all this, of course, is costly, and we don't yet know if it can be doable within the next years to implement it routinely in the selection and conservation programs. Of course, technology is improving and the costs are decreasing, but yeah, we are not yet ready to have this done at a broad scale in Switzerland. So this needs still efforts, but I think this is the direction where all different countries are going. There are some new genomic tools which are developing. So at the end, I think this will be a broad approach. So I hope I have succeeded to give you an overview of our current activities here at Agroscope regarding the dark honeybee. So this is not my own work. It has been done in collaboration with many partners from France and Switzerland. So Annick Gmel has been in charge of the part about genetic diversity. So her paper will come out hopefully quite soon. The genome-wide association studies, one is published, one is about to come. So I hope you will have a look to our papers. And thank you for the Federal Office for Agriculture, which is providing funding for this type of research. And I also thank you for your attention and CCAM for inviting me to this meeting.
Right. Thank you.